Good afternoon, everyone. Thanks for sticking around. Thanks, Dr. Chabner, for the invitation. Taking a step back from the front lines of infectious diseases and bone marrow transplant, from pediatric ALL to the underpinning of all of this science, and the chemistry is where I hang my hat and where I do my work. I have nothing to disclose. Molecular imaging is something that was on my mind this morning as we heard about the fascinating and complex transformations that are underlying our treatment of lung cancer. And the more we know about cancer biology, the more intricacies there are that are really playing out below our radar. And we do biopsies. Jeff and Leisha and Alice have been following up at discrete time points. But on a day-to-day basis, our clinical tools are really pretty primitive. And we use X-rays and CT scans to take an initial stock of our patients and follow our progress with treatment. And at our best, we can go to the fourth dimension by adding in metabolism. Really, PET scans give us color in our own visual interpretation. But it's a four-dimensional scan in a way. It's not a time series, but it's an added layer about the glucose utilization in a tumor. And so you might imagine, if I can hit the laser here without hitting the back button, that the underpinnings of all the oncologic signaling, the chromatin remodeling, the immunobiology, are all happening on a day-to-day basis. And we might learn a tremendous amount if we had better tools for seeing them happen, not just snapshots at the time of relapse, not just wondering how the cocktail of therapy that we were inventing this morning for lung cancer is playing out, but actually tuning it in real time. So all of that's impossible right now because molecular imaging is really hard. And so I could give an hour-long talk on why it's difficult to develop probes that have contrast, that have signal, that have specific targeting of the pathology that you can see on the outside of the body that work in a human, not a mouse. And superficial versus deep detection, as an example, macroscopic versus microscopic scale. How many positrons do you really need, and how small an object can you see them when they're coming from a CT scan representation of a body? Background is really a simpler thing to imagine than all of the physics of these other topics that I'm glossing over, which is basically that our probes, at their best, accumulate where we want them to, but to a lesser degree everywhere else. And so we've seen PET scans in a variety of clinical series today, and they all have a lovely pinkish sunset glow with a brighter yellow spot superimposed on it. And so pushing the boundaries of molecular imaging, we need to get rid of the sunset glow so we can see tinier yellow spots. We need to get rid of the background in order to be able to see or to imagine, since this is a prospective methodology development, kind of a chemistry talk. In order to be able to imagine that smaller scale, the sunset has to go away. And so here are those PET scan images I've just been describing. And here's a big lung tumor, which we can pick out right next to the heart. The heart's brighter than the tumor. And wouldn't it be great if PET scan were really only the beginning? So in order to do that, we can't do it right now with the drugs we have, with the proteins we have, with the antibodies we have. But there's some hope, and I'm going to tell you about my own daydreaming in this regard today, that some of the novel chemical tools, the click chemistry that got apostrophes in the title of my talk, could be used at a beginning step in devising nanomachinery that can go into a living organism and specifically isolate an underlying biological process of interest to transplant, to treatment, to all of these fascinating hem-onc topics. So what the devil is click chemistry? It's a field that was invented by a chemist after he won his Nobel Prize. And he said, what else am I going to do next? And his daydream was that instead of needing solvents and glassware in carefully controlled conditions, that you could sit down ab initio and design reagents that were so good that if the two pieces came together anytime, anywhere, you'd get the product you wanted. And it was widely reviled by serious chemists because that's too easy. You won't be able to make interesting molecules with such simplistic chemical concepts. But where that criticism maybe missed the mark is that you're not going to synthesize the new lead compound at Epizyme with click chemistry, but you might be able to make a toolkit that can do chemistry in spite of everything else that's happening. So you won't make the stereocenters, you won't make the exquisite SAR-defined structures for sub-nanomolar potency, but you might be able to take pieces and have them do interesting things. So I've just put together a quick schematic here. So A plus B, they should stick. And they should do it anytime, anywhere, no matter what else is around. So this chemical toolkit shouldn't care about proteins, salts, DNA, RNA, sugars. The entire biological milieu should be transparent. And you should be able to do it with these pieces attached to biomolecules. And this is really the power of the idea that I've been hypothesizing about and spending my time in the lab working on, is that if we're smart enough about what we attach these post-Nobel reagents to, we should be able to make the big things do interesting tricks for us, even if the chemistry that's at the core of the reaction is just a one-trick pony, or whatever the serious organic chemists might have described it at after the talk. So the particular click chemistry that I use, it's a bioorthogonal chemistry. I'm remembering my own slides, I put this definition at the top. Bioorthogonal is another phrase that you might come across in the decade ahead, if we have any luck, is another phrase to describe click chemistry, which is to say that all of these reactions are perpendicular to the chemistry that biology uses. The reagents don't care about the biological molecules, and the biological molecules ignore the reagents, the green and the blue molecules that I labeled as A and B. So if we're going to do synthetic chemistry in living systems, take advantage of your slide, you'd want a pair that did all of this really effectively. You'd want it to be able to work at the nanomolar concentrations. You'd want it to be able to function without catalysts. We can't deliver copper or palladium or microwaves, any kind of chemical tools into a living organism or into a living cell. And so it should happen quickly. It should happen in all these technical ways with minimal intervention. And so tetrazine and TCO, so if you hear me say TZ or TZ, TCO, that's the pair that I've been working with in the Weiss-Slater lab here in systems biology. So tetrazine is a really cool molecule, and my mentor calls these spring-loaded weapons. That's his synopsis for how these work. And I'll gloss over all of the fascinating chemical physics of why it happens and just stick to the basics, which are that this is the tetrazine in the box, and that we've got a handle on it that we can use to attach to fluorescent tags, to nanoparticles, to other drugs, to radio labels. And so it really is the delivery vehicle that that initial hemispherical cartoon might have indicated. And its partner, TCO, is an 8-membered ring that's been wrapped in a corset. The bond is kinked. It puts all this spring-loaded strain into the ring. And that's the easiest way to just analogize the magic trick of what makes this particular pair so effective at finding each other and reacting so spectacularly quickly and with facility. And so TCO also has a handle on it. We can attach it to DNA, RNA, nanoparticles, you name it. And this is a 2-scale drawing of what a TCO tag looks like on an antibody. And so again, this is all schematic background for you, but a TCO tag is tiny relative to the scale of a biomolecule. And the two things I wanted to point out here is that you can imagine that this is a relatively small footprint. The tag needs to be as small as or smaller than whatever it is you're using it to label. And that this is color-coded in yellow to show all of the lysines that are on this particular antibody crystal structure. So there are tons of them. And so because it's so tiny, you can imagine that there's an inherent amplification capability here, that we could put lots of these tiny tags onto something like an antibody and amplify the signal. So think about the way we do that traditionally in primary antibodies, secondary antibodies. Here there's a chemical amplification strategy. So when you put these two pieces together, I've got a rotating video here so you can see the corseted TCO, and then the product when the two pieces have stuck together. They reliably do just that hypothesized A plus B goes to a covalently linked, chemically stable. It's not a non-covalent interaction. This is just permanent connection between the two pair. So tiny tags, I alluded to that when I had my antibody scale drawing up there. You could tag a drug molecule with a TCO. It's smaller than a drug molecule. This chemistry works in vivo. It satisfies all of the criteria of chemical orthogonality. It doesn't care about DNA, doesn't care about proteins or water or salt. You can imagine schemes that I won't take time to talk about today in which we can label things in one step, let them do their thing, let the kinetic process play out, and then detect them in a separate step, because the chemical reaction can go find its partner, wherever that is, hiding in cells, hiding in the nucleus, hiding in the proteome. And you can do it in vivo. So you can imagine a whole new generation of assays where instead of extracting and western blotting, northern blotting, southern blotting sequencing, with this kind of pairing, you can do chemically amplified, covalently tagged magic tricks in situ at its best in our prospective vision of it. And coming back to molecular imaging, you can imagine that if you have the right scheme, you can use this to reduce background signal. You can let clearance processes play out and then detect the result. You can, and I'll go into this more as we go forward. So background signal, which I started with, is that sunset analogy in the PET scan, is this quintessential challenge in molecular imaging. So starting the story of the work that happened in the lab upstairs here on the 5th floor, It was noted a couple years ago by one of my predecessors that fluorescent molecules can be quenched. They can be turned off so that they don't emit light anymore by attaching tetrazine to it. And you'll see that I've color-coded tetrazine here. It's blue through all these slides. And so if you just tag a fluorophore, which is I've given the ghostly white color here with the tetrazine, an energy transfer process, the physics of which I'll gloss over, makes that molecule non-fluorescent. And happily, if you take away the tetrazine by reacting it with TCO, which it's so eager to do, the fluorophore becomes fluorescent again. And so this is a signal amplification strategy, a fluorogenic process or fluorogenic ligand, where we've taken advantage of the tetrazine's reactivity and we've given a visual readout or a fluorescent readout to it. And so this particular pair, in the work from a few years ago, the molecules became 20 times brighter after they reacted, and with a 20 times signal amplification of the bright molecule after it had stuck to its target, in this case Taxol on microtubules in a cell, you can see an image of something hazy resembling microtubules emerge from the background. So what I set out to do was to figure out a way to make this work better, because I was interested in these click chemistry tools and the bioorthogonal imaging and having molecules that you can send off on their way to find their target. And so I explored a series with a colleague of mine, another chemist in the lab, of ways to connect a tetrazine onto a fluorescent molecule to make this light switch effect work a lot better. And so there's another theoretical underpinning of which the data was too difficult to summarize and encapsulate for general interest, and so I've happily omitted it for all of us. But by wiring the molecules together, and I've just represented it in a slightly different space-filling form here, but by wiring the pieces together, a fluorescent dye with a tetrazine just right, you can propose exceptionally efficient energy transfer, exceptionally efficient switching. And it turns out that this hypothesis worked pretty well. The molecules actually get 1,500 times brighter when the tetrazine reacts instead of just 20 times brighter. So what does that look like? So if you start out, one of these two vials has the fluorophore in it under UV irradiation, and the other one's just water. And to my own eyes, I can't see the difference between the two. Although beforehand, there's absolutely no detectable fluorescence. It's just a clear solution. Afterwards, it becomes intensely, brightly, greenly fluorescent, like the dye should have been all along. So immunohistochemistry, immunofluorescence, all of the images you can imagine microscopically, this is the same sort of switching on effect. Instead of always being bright, this molecule is only bright after the tetrazine is clicked to its TCO counterpart. So here's just a data point, the fluorescent spectrum before and after. It's more than 1,000 times brighter than it was before. So now what about imaging? This isn't a physics seminar, right? We need back to our cancer cells, back to an application. And so starting with an EGFR overexpressing cell line and saying, let's set out to image this by adding tetrazine dye to a TCO-labeled field. You can imagine making cetuximab with TCO, well known to everyone here. And whereas if you add the click dye to a control antibody, you see nothing, and this is just at a high concentration of dye, it should look like this green square if it weren't so beautifully quenched by the magic trick that I've elided. If you add that same dye, the same amount of dye, to fixed cells that have EGFR TCO on them, anti-EGFR TCO or cetuximab TCO, with no washing and no rinsing, it shows up better on our side flanking monitors, I think, than it does on the big central screen. An image emerges from the background which corresponds beautifully to just where we'd have expected the EGFR to be. If you add it to live cells, happily the exact same thing happens. The dye is sitting there at full test, full strength, but only where it binds to its targets that we've pre-parked onto the surface of those receptors does the fluorescent signal emerge. And so if you say, well, how does this compare? This is scrambled here as well. I'm sorry to see that. How does this compare to the first generation probe? The 20 and the 1,000 are still working. For an intracellular target, you can see that the nanoparticles that these macrophages have ingested are pre-labeled in red. And the click dye, as we've dubbed these things, recapitulates the location of the nanoparticles beautifully. I could point over to the TV screen, it shows up even better. And the traditional first generation molecule does the same thing, but with this very high background that would prevent you from doing any subtle oncobiology analyses. And if you merge those two images, you can see a beautiful yellow image showing the co-registration that the dye has found the TCO target where we've left it. So you can look at other things. You can look at actin, labeled with probes that have like fluoidin TCO, a small molecule that binds to actin and adding the dye out of the darkness, once again, a specific high signal-to-noise ratio image emerges. You can zoom in further and you can see the subcellular structure of those filaments. Again, no washing, no rinsing. And I might have pointed out that labeling things in vivo, of course, we can't wash them, we can't rinse them, we can't do the pathology labs, rigorous 2 to 4 hour washing steps that they do for every IHC that they report back to us on our biopsies. And so this kind of a labeling routine, although complicated in construction, opens the doorway to imagination about how you can deploy these same chemical tools in model organisms and living cells, and eventually, as the stars align and as technology marches onward, possibly in people. So intravitally, I've lost track of exactly how much time I have, but you can deploy these same tools into a mouse. And in the lab before I started, there have been a whole host of tetrazine-TCO experiments done for in vivo diagnostics analytics. And so here in the pre-ejection state, we can see that a mouse that's been injected with cetuximab TCO looking at a window chamber tumor model. Here at the top right, there's no fluorescence in the window chamber. And after we add the dye, lo and behold, the same cell line that we could do in a trivial format in glassware, we can generate the same sort of relatively high signal-to-noise target-specific imaging. And we've gone on to characterize that further in some other model systems where we've taken advantage of endogenous labeling to pre-label the target. So here the membrane of the cancer cells has been pre-labeled with red fluorescent protein. And again, on addition of the dye intravenously, the molecules diffuse, they transmit through the bloodstream, pharmacology happens, they find their target, and out of the background emerges a fluorescent image. We've extended that to blue, so we've got a whole host of new probes that we're working on, multicolor being the name of the game, and flow cytometry and immunopathology, multiple targets, multiple colors. And historical approaches to try to turn coumarin dyes, or blue dyes, on and off can achieve 3-fold or 60-fold turn on. And our new results, exciting, we can hit numbers like 7,000-fold brighter, which is almost a ratio that's too big to conceptualize what that means exactly for a molecule to turn from that dark to that bright. And we've shown that these new blue probes can be used to generate the same sorts of click images that we've done with the green dyes that we started this off on. So going forward, I've told you a little bit about the concept of click chemistry, bioorthogonal chemistry, and this particular pair, tetrazine and TCO, that works so well to pull off the original vision of anytime, anywhere reactivity. And obviously this fluorogenic molecule that we've discovered and spent a lot of time working out the chemistry and the photophysics of suggests to us some applications for cancer imaging, for molecular imaging, as I've introduced in just our simple demonstration proof of concept experiments. So we're working actively on taking this methodology and turning it into a well-honed data set and well-honed set of tools. I've talked about how this can be used for small molecule labeling, and a proof of concept with an antibody is just the beginning. So irreversible EGFR inhibitors, we can attach TCO. BTK inhibitors, attach TCO. And in this two-step labeling system, you can imagine deploying these molecules into a whole host of biological contexts. And mouse models will be the next step as these projects continue to succeed with a bit of good fortune. So we're working in the lab, my colleagues and I, on a whole host of projects that apply this molecular pair of tetrazine and TCO, and click chemistry, I think, is increasingly capable and likely to rear its head in oncology going forward, which is part of the rationale I had for introducing you to a bit of it. So we're working on second-generation dyes that are brighter in vivo and in the aqueous systems than what we've started with. We're working on super-resolution imaging and on tools to actually do serious biology that would be useful for the click chemistry as a probe of biomolecular interactions in situ, not just labeling experiments with dark to bright. So thanks to everyone, and I'm happy to take any questions.
On one of your first, you had two proteins, or A and B. They were, one had your two small molecules,
Yeah.
and then they click together. Has that been implemented to visualize molecular interactions between proteins, or could there be some problems with steric relationships where the proteins had to come together in the right way to allow those two molecules to click.
Yeah. Other work in the field has shown unnatural amino acid applications with these, where model organisms can synthesize proteins that have specifically incorporated tags at the right places. So when George Church finishes editing the genome for everyone to be able to do this with greater facility, you can totally imagine encoding proteins for 2 hybrid assays, 3 hybrid assays with a covalent readout like this. Or even having the organism incorporate just one of the tags and then using bimolecular or bivalent tetrazine chemistry to tie them together with a staple, so to speak.
Bruce? Is it possible, Jonathan, to imagine just giving the antibody first, letting it wash out from every place but where it's, tight binding, and then giving a ligand with a radio labeled or radio nuclide on it?
Yeah, absolutely. One of the challenge. Yep, absolutely. So FDG is tricky to work with because it has to be imaged quickly, and antibodies and FDG don't mix well for just that reason. So one of the applications that we can certainly imagine is the 2-step approach of a slow phase of targeting and a second phase of readout or therapeutic delivery. That's a click chemistry application waiting to happen.
Have you done any in vivo studies, or is that mostly
so-
in vitro so far?
So we've done the mouse imaging experiments where the two-step labeling process works in the window chamber models of human tumor cells implanted into the mice. We haven't yet done imaging of endogenous mouse targets, for example, but I think that's just around the corner. And it would actually make for a better system than the somewhat labor-intensive process of implanting the hardware and waiting for the human cells to grow, to actually look at the mouse biology for
Okay, thank you very much.